Good afternoon, everyone. Please take a seat. So my name is Harry Sokol. I'm professor of gastroenterology in Sorbonne University, Saint-Antoine Hospital. I will moderate this afternoon session, and I will also give the first talk. Okay. So let's start so we'll not be too late. Oh, it's working. Yeah. Okay, so as I told you, I'm professor of gastroenterology in Sorbonne University. I'm also a co-founder of a startup called DIM BioConvS. And basically, I'm going to describe you our journey from the lab discovery to the first clinical trial with a bacteria called Faecalibacterium prausnitzii, which is derived from the human gut. So as I said, I'm a co-founder of Faecalibacterium, so that's my main conflict of interest for this talk. So just a few reminders, just to set the scene. In our gut, we have around 2 kilograms of intestinal content. So this is not only bacteria, there is a lot of water in these 2 kilograms. We estimate currently that we have as many bacteria in the gut as the number of cells in our body, so roughly 10 to the 13. And in 1 gram of stool, we have more than 100 trillions of microorganisms. So in our cells, we have obviously the human genome, and if everything is fine, it's the same human genome in all our cells. If you put together all the genes present in the microbiome of one healthy subject, it represents roughly 25-fold the human genomes, meaning two things. First, that if you want to study the gut microbiome in depth, you are going to need a lot of sequencing, a lot of data, a lot of storage, and also computational, you will have high computational needs. And it also means that there is a huge theoretical metabolic potential in the microbiome. So in our microbiome, we have bacteria. This is at least the part we know the best. And in the colon particularly, most of the bacteria are strictly anaerobes. They are belonging to these two phyla, the Bacteroidetes and the Firmicutes. But we also know now that there are many other types of microorganisms, fungi, viruses, and particularly phages that are targeting the bacteria, and even other types of microorganisms such as archaea and protists. So this was just basically the introduction. Now I'm going to dive into the topic itself. I will talk about the interest of this bacteria, Faecalibacterium prausnitzii, in inflammatory bowel disease, and then in immuno-oncology field. So what are inflammatory bowel disease, or IBD? They are conditions characterized by chronic inflammation in the gut. There are two types of IBD, ulcerative colitis and Crohn's disease. They are affecting young subjects, altering their quality of life. They are chronic disease with flare and remission. Unfortunately, we do not have curative treatment, only treatments that are able to basically control the symptoms and the inflammation. And the patient can undergo different types of complications, severe flare leading to hospitalization, but also infection or neoplasias that are related either to the intestinal inflammation process itself or favored by the immunosuppressant we have to give to these patients. So these diseases are not rare. The incidence in France is about 10 for 100,000 per year, and it's roughly similar in all Western countries. And we have currently around 300,000 patients with IBD in France, and the long life cumulative risk to develop one of these diseases is beyond 1%, and these numbers are increasing every year, both in westernized countries and also in countries that are basically acquiring our way of life. So the pathogenesis of IBD is not clearly defined, but if you read any review or any book chapter on this topic, you will probably find a sentence like this one telling that these diseases are related to an activation of the gastrointestinal immune system toward the gut microbiome in genetically susceptible hosts and under the influence of environmental factors, meaning that these diseases are complex, obviously, and there are all these actors. But in the last 15 years, I would say, there are now more and more data suggesting that some of the environmental factors, some of the genetic factors are going to impact the disease through an effect on the gut microbiome. Of course, it does not mean that everything goes through the gut microbiome, and many other factors impact the disease independently of the microbiome. So I'm telling you that the microbiome is involved in this disease, but you don't have to believe me. Do we really have proof that the gut microbiome is involved in the pathogenesis? So I think that now, and this is mostly related to the data that have been generated in the last 5 to 6 years, we have a pragmatic answer to this question, and the pragmatic answer is yes. There is a long list of arguments, but I'm going just to mention the ones that are the most convincing, at least from my point of view. First, we know that modifying the gut microbiome impacts the disease severity. This is true in mouse model of inflammation. This is, okay, nice, but obviously this is not the most important. What is important is that it's also true in humans, and this is probably one of the most crucial contribution of fecal microbiota transplantation in clinical trial is to show that in humans, when you modify the microbiome, you have consequences on the disease. Second important argument is that actually the microbiome from patients with IBD exhibit pro-inflammatory effect. And if you transfer it into germ-free mice, the mice will have a higher level of intestinal inflammation at baseline, and they will be also more susceptible to inflammation compared to mice colonized with a normal, healthy human gut microbiome. Of course, the last argument, which is actually the beginning of the story, is that the gut microbiome is altered in patients with IBD. If you analyze the microbiome of patients with IBD, you will see alteration. So this is one of probably several hundreds of studies that has been published now showing the alteration of the gut microbiome in patients with IBD. Here, each dot represents the microbiome of one subject. If 2 dots are close, it means that the microbiome are similar. If 2 dots are far on this figure, it means that they are different. And here, it's not magic. You can very easily see that the healthy subjects in blue have a different microbiome compared to patients with IBD in flare in red. And in green, you have patients with IBD in remission, so without active inflammation, and you can see that they are in between. So the microbiome is different. Another characteristic of the alteration of the microbiome in IBD is that its diversity is decreased, and this is really the same type of diversity that you use when you are describing ecology in forest or in ocean. It's also a reduced number of different species and also an alteration in the balance between the species. In terms of composition, the microbiome is different, but what are the characteristics of these differences? So there are many types of differences, but the most reproduced one and striking one are that first, compared to the healthy subjects, which are dominated by Firmicutes and Bacteroidetes, the 2 big groups of bacteria I mentioned at the beginning, here in patients with IBD, you have a decrease in the Firmicutes, which is the blue group here, and you have a bloom of the orange group, which are the proteobacteria, and in the proteobacteria, you have Escherichia coli, for example, but also Klebsiella and others. And actually, one of the strongest signal that has been found and repeatedly found everywhere in the world is that patients with IBD have this increase in proteobacteria and particularly of E. coli. And among the Firmicutes that are decreased, Faecalibacterium prausnitzii is also a very strong signal that go down. And actually this has been, as I said, reproduced everywhere in the world. An interesting piece of data that has been published very recently, a few months ago, is that actually the alterations that are observed in the gut microbiota of patients with IBD, and particularly Crohn's disease, actually precede the diagnosis by several years, meaning that up to 5 years before the diagnosis, when there is still no inflammation, no symptoms, you already start to see the alteration in the microbiome, including the decrease in Faecalibacterium prausnitzii. So our story with this bacteria started with this paper in 2008. We observed that basically in patients with Crohn's disease, patients who are undergoing ileocecal resection, which is the type of surgery which is very frequent in Crohn's disease. In these patients, the lower is the abundance of these bacteria, the higher is the risk of recurrence. So there is basically a negative correlation between the abundance of the bacteria and the risk of disease recurrence. And we also observed similar results in another setting. So these are patients with Crohn's disease again, treated with anti-TNF agents, so quite powerful immunosuppressant. And in patients who are in remission and who stop this treatment, the level of the bacteria also predicts the risk of recurrence. And again, if you have a low level of the bacteria, your risk of recurrence is higher. So this is obviously correlation. And in terms of alteration of the gut microbiome, as I said, we and others found this decrease in Faecalibacterium prausnitzii, and actually there are other types of bacteria that are decreased. And others also focus their work more on the pro-inflammatory bacteria, which are increased in IBD, such as E. coli. So based on all this data, we hypothesized that indeed Faecalibacterium prausnitzii may have anti-inflammatory effect. Again, it's decreased in patients. The lower is the abundance, the higher is the risk to have a flare. And so we tested this hypothesis also because we knew that this bacteria is actually very unique in the gut microbiome. First, it's the most abundant species in the gut, meaning that by itself it represents around 5% of the gut microbiome alone. And another characteristic is that it's highly prevalent, and this is what we call the core microbiome. It means that this bacteria is actually present in almost every healthy human subject on Earth, and this is quite a rare characteristic also. And so this is the result of our hypothesis. Actually, we demonstrated that both in vitro, in different models, and also in vivo, these bacteria exhibit anti-inflammatory effect, particularly regarding intestinal inflammation. And actually, this paper that has been published more than 15 years ago now, actually was quite new at this time. It was basically the first time that the concept of using a bacteria from the gut microbiome that was selected based on observation in human was used as a potential therapy. And this basically led to this concept of live biotherapeutic products, which is to use bacteria from the microbiome as a drug. So, as I said, this bacteria is very specific. It has all these characteristics that I already mentioned. It has also this immunomodulatory effect. And based on this effect, we thought there was some therapeutic potential with this bacteria. And of course, with academic funding, at one point we were stuck. It was not possible to find enough funding to develop the manufacturing, to do the clinical trials that are very expensive to do. And so that's the main reason why we funded Excellium Biosciences with Philippe Langella, who is a microbiologist in INRA, and also with Patrick Gervais, who is a specialist in production process. And we found the last piece that we really needed was this business and biotech expertise. And it's Benjamin Adida, we also funded the company with us, who took this hat of CEO, and obviously he brought all his expertise in business development and biotech that obviously we did not have. So we founded the company in 2016 with a first part of Series A, and then we focused on the first clinical trial, and the idea was really to try to do this clinical trial as fast as possible, but there were actually many obstacles. First, the manufacturing was a huge obstacle because this bacteria is not an E. coli, it's not a lactobacillus, it's very difficult to grow. It's extremely sensitive to oxygen. It grows usually in the lab in very rich culture media, so we had to solve all these issues before being able to even think about testing it in humans. The other difficulty was that at this time there was no clear regulatory path to develop this type of drug. And so basically we had to co-construct the regulatory path with several European agencies. We also had to think a little bit outside of the box because this is a bacteria we are using, an immunomodulatory bacteria. This is not a powerful immunosuppressive drugs. And of course, it's nice it's not a powerful immunosuppressive drugs because this is what we want to try to avoid or to decrease. All the side effects that are associated with these immunosuppressive capabilities. And so we had to think about the design in an innovative way. And we have to act on new targets. And the interesting aspect also was the strong motivation from the patients. And so we started our study at the end of 2022. And of course, in the meantime, we worked also on the mechanism of action, and we deciphered actually several of the mechanisms of action involved in the anti-inflammatory effect. We showed that indeed there is some molecules that are produced by the bacteria that are notably acting on epithelial cells by reinforcing the intestinal barrier. We also showed that there is some component on the bacterial wall that are acting on immune cells, on antigen-presenting cells, most importantly, and that are changing the immune phenotype of these cells toward an immunoregulatory profile with notably a strong production of IL-10. More recently, we showed, and this is still a paper which is submitted, that a large part of this anti-inflammatory effect in antigen-presenting cells is mediated by a complete rewiring of the metabolism and notably the energy metabolism of these antigen-presenting cells. So, as I said, we did this first study, this first in-human study. It was a phase 1/2A. So directly in patients with Crohn's disease, so we didn't have to go through the healthy patients route, the healthy subjects route. 8 patients were enrolled. One originality also of this study was that we treated the patient for 6 months. So again, for a first-in-man study, this is quite unusual. We didn't have any safety signal, and actually we just start now the analysis of the results. We just received in the lab the samples actually yesterday. So we are working on it to try to understand and to maybe to find some target engagement in humans. In the meantime, we are working on a phase 2 study that actually is going to start in the next few months, and this will be a randomized and controlled study in post-operative setting, also in Crohn's disease, with the help of the REMIND consortium. So second aspect, which is immuno-oncology. So, I mean, you all know how immune checkpoint inhibitors are working. It basically unleashes the immune system towards the tumor. And as the gut microbiome is involved in the immunomodulatory effect, very early, several groups thought that the microbiome may modulate the effect of immune checkpoint inhibitors. And this has been actually shown by the team of Laurent Zidvogel in 2007, both in mouse model, showing that antibiotics will basically decrease the efficacy of immune checkpoint inhibitors, and also in some retrospective human study, showing that basically patients taking antibiotics had a lower efficacy of immune checkpoint inhibitors. So Very rapidly, the first question was to know whether some bacteria, specific bacteria, were associated with this phenotype. And quite surprisingly, only few bacteria popped up among the first studies that has been published. And one of these bacteria that came in several studies was, again, Faecalibacterium prausnitzii. And this is one of the early studies in science from a US group in melanoma, showing that the Faecalibacterium prausnitzii signal was the strongest to predict a response to immune checkpoint inhibitor. And then this was confirmed by many others. This is a meta-analysis in melanoma. These are two other studies in non-small cell lung cancer and hepatocarcinoma. And here, this is a recent study, again, from the team of Laurent Zidvogel, who built and validated a microbiome-based score to predict response to immune checkpoint inhibitors across a variety of cancer. And among the most prevalent taxa in this signature, again, we found Faecalibacterium prausnitzii. So before entering this field, we wanted to check ourselves if really the signal was there in humans. And so we had access to this study, which was performed by the team of Laurent Zidvogel. In this paper, they focused really on Akkermansia muciniphylla, which is another bacteria which is known to activate or to improve the response to immune checkpoint inhibitors. And so in this study, they showed that basically patients without Akkermansia had a lower response to immune checkpoint inhibitors. So we got access to this data thanks to Laurent Zidvogel and Nicolas Segata, and we reanalyzed the data with a focus on Faecalibacterium prausnitzii. We separated the cohort in 3 parts, and basically what you can see here is that patients with high abundance of the bacteria had a much better response to the drug in terms of overall survival. And interestingly, when we looked at the different types of patients, notably in this study, there was both first-line patients and second-line treatment. The signal was found only in patients in second line and not really in first line. Another aspect which was of particular interest for us is that patients who were treated with antibiotics, actually, in these patients, we did not find any signal with Faecalibacterium prausnitzii, suggesting that indeed, of course, if you perturbate the microbiome with antibiotics, you completely lose the predictivity of these bacteria, and this means that it suggests at least that there is some kind of causal effect between the microbiome and the response to immune checkpoint inhibitors. And when we did a multivariate analysis to take into account all the potential confounding factors, which are known to impact the response to immune checkpoint inhibitors in this cancer, actually the effect of Faecalibacterium prausnitzii was even higher, with a delta in mortality reduction of more than 40% compared to patients with high to low Faecalibacterium prausnitzii. Then we generated also our own data. So this is a study in advanced gastric adenocarcinoma. We had access to the sample of this cohort, and we did the metagenomics and the analysis ourselves for this one. And again, what we found here in terms of overall survival, we found different signals. We found some bacteria that were associated with an increased mortality, such as Veillonella, for example. And these bacteria are known to be associated with other cancer types. And we found some bacteria, such as Enterocloster, which is already known to be of bad prognosis in immune checkpoint inhibitor treatment. And again, we found Faecalibacterium prausnitzii, which was associated with a decreased mortality. And interestingly, when we looked at progression-free survival, which is the other common outcome we look at in cancer, Faecalibacterium prausnitzii was the only one statistically associated with a longer progression-free survival compared to the other bacteria. So, of course, here we see correlation, comparison, but is there a causal link, or is it only a pure biomarker? So, actually, this question was present very early on, and already in this study, I already mentioned This group from the US tried to address this point by colonizing germ-free mice with the microbiota from responder or non-responder to immune checkpoint inhibitors. Then they injected the mice with melanoma cells, and then they treated the mice with immune checkpoint inhibitors. And basically, they observed that indeed, mice colonized with the microbiome from responders had a better response to immune checkpoint inhibitors compared to the mice colonized with the microbiome of non-responders. And already here, they also found this link with Faecalibacterium prausnitzii. So the question now is, what can we do for all these patients with low amount of Faecalibacterium prausnitzii and altered response to immune checkpoint inhibitors? Before starting our own clinical trial, we wanted some clues, and so at this time, these two studies were published. These are fecal microbiota transplantation studies in patients who basically failed immune checkpoint inhibitors. And so they got FMT, and in the first study, 3 out of 10 patients gained response to immune checkpoint inhibitors, and in the second one, 6 out of 15, gained response to the treatment. And interestingly, again, our bacteria was enriched in post-FMT microbiota of responders, suggesting again a link between the microbiome transfer, the response, and the bacteria. So, of course, now the question is to know whether the bacteria itself can improve the response to immune checkpoint inhibitors. To make a long story short, we did some experiments in vivo in mice in a very common model of very common tumor-bearing model mice. We did some alteration of the microbiome with some mild antibiotic treatment. The idea was not to wash out the microbiome, it's just to induce some kind of perturbation. And then the mice were treated or not with Faecalibacterium prausnitzii strain EXL01. And so this is the tumor growth. You can see in red, compared to the gray, which is a vehicle, that anti-PD-L1 is working. The tumor growth is decreasing. You can see that with antibiotics, actually, you decrease the efficacy of immune checkpoint inhibitors. And when on top of antibiotics, you bring back these bacteria, you basically regain the response. And this is even nicer when you look at survival. You can see here that the antibiotics is basically reducing the efficacy of the immune checkpoint inhibitors. And when you bring back the bacteria, you bring back the efficacy. So we did some RNA sequencing to understand a little bit better how it works. And just to give you a snapshot of the result, if you just look at the differentially expressed genes between no treatment and anti-PD-L1 treated mice, you see that around 4,000 genes were differentially expressed. If you look at the effect of the antibiotics, you see that there are more than 3,000 genes that are differentially expressed. If you look at the effect of the bacteria, so EXL01, you have more than 4,000 genes that are differentially expressed in the tumor. And then, and this is probably the most interesting part, if you compare the mice treated with anti-PD-L1 alone to the mice treated with antibiotics, anti-PD-L1 and EXL01, then you almost do not see any differences, suggesting that the alteration of the microbiome induced by the antibiotics has been completely restored by the bacteria. And for sake of time, I'm going to pass this part. We did some in vitro experiment as well in human cells and showed that indeed the bacteria itself stimulate the production of gamma interferon of T cells, increase the activation marker of antigen-presenting cells, and more importantly, in this mixed lymphocyte reaction assay, of course, all of this was done in presence of immune checkpoint inhibitors. You can see that the more bacteria you add in the system, the higher is the production of interferon gamma, and you also have some small signal regarding proliferation of T cells. So the mechanism of action are still under investigation for the immuno-oncology aspect. It involved different things, effect on disease, and we think that really the antigen-presenting cells are really important in the mechanism of action, maybe regulation of PD-L1 expression, and also it involved boost in T-cell function. So, in conclusion, for immuno-oncology, the gut microbiome is involved in efficacy of immune checkpoint inhibitors. We saw and confirmed ourselves that high level of Faecalibacterium prausnitzii at baseline is associated with better response in multiple types of cancer. If you use this bacteria in animal model in vivo, you can see that it boosts efficacy of immune checkpoint inhibitor. And obviously, the next step was to evaluate the effect in clinical trial. And we have currently 3 ongoing clinical trials, 1 in gastric adenocarcinoma, 1 in non-small cell lung cancer, and 1 in hepatocarcinoma. And regarding IBD, we have to again keep in mind that when you are using a bacteria as a drug, it's not an immunosuppressant, it's an immunoregulatory agent. And so we have to shift our mindset regarding the use of this type of drug. We have, and so just to summarize all the clinical trials that are now ongoing, I mentioned these 3 clinical trials in immuno-oncology. We hope to start soon a 4th one in renal cell carcinoma in the US. We have 1 Crohn's disease study that should start early next year. This is a post-operative study I mentioned. We are in the process of finalizing the regulatory aspect for this study. And I did not mention it because of time, but we are also using this bacteria to try to replace fecal microbiota transplantation in recurrent C. difficile infection. And we have a study also ongoing on this aspect. Thank you very much. Is there any question?
Thank you so much for this great talk. I have a question about the mechanism of action. Do you think that by somehow extracellular vesicles may be related to part of the mechanism of actions of FPRO?
I mean, we don't have definitive answer, but what I can tell you is that in vitro, we don't think it is because we are using washed bacteria. So we are not using the supernatant. And basically, when we use the bacteria in vitro, they are exposed to oxygen, so we don't think there is production of this vesicle. But in vivo, who knows? Yeah.
Hey, great talk. I guess my question relates a little bit. I was wondering, in all the effects that you showed, which ones require live bacteria, or which one can also be elicited by the dead bacteria? And in all the trials you're conducting, is it live bacteria that you're delivering to the patients?
Yeah. So what we know, at least in vitro setting, I mean, first thing, if you gavage this bacteria into a mouse, in the stomach of the mouse, our simulation, I mean, we simulated that in vitro, the bacteria are not metabolically active anymore, meaning that they are not going to grow again. Obviously, there is tons of oxygen in the stomach, there is low pH and all of that. The bacteria are not anymore metabolically active, but we still see the effect both for cancer and inflammation. So this is suggesting that, again, in vivo in mice, the effects are mostly related to molecules present on the bacterial wall than on molecules that are produced by the bacteria. In vitro, if we take the supernatant and use it to treat cells, we also see some effect, notably on epithelial cells. So if in vivo, in humans particularly, the bacteria is able to produce these molecules, we think it would be also beneficial. And to answer your second question, yes, the bacteria are alive. The production, all the manufacturing is actually a nightmare because we want to keep it alive. So all the production is in anaerobic condition. And the bacteria are given alive to the patients.
Yes. Are they strictly located into the intestine tract, or do they actually migrate into lymphoid organs, perhaps even in the circulation?
Yeah. So what we think is that the bacteria stay in the lumen, at least as a full and living bacteria. Of course, you see components of the microbes, of the gut microbes in lymph nodes. You see components in the blood. If you look for DNA, microbial DNA in the blood, you will find it. But it's not live bacteria. So there have been, all this discussion about blood microbiome and all these things. Most of microbiome experts think this does not exist. What we have in the blood, in lymph nodes, except in pathological condition, is a fragment of microorganism, microvesicle. Yes, that's very likely, but not live bacteria.
And what effect do they have directly on cells of the immune system, like in macrophages?
Yes, so we see that basically they are changing completely the polarization of the macrophages. Basically, they are, even it's a little bit oversimplistic, they are pushing the bacteria toward a kind of M2 phenotype. And we see that also at the metabolic level. And actually, in our last experiments and the papers that is submitted, we show that if you block the rewiring of the energy metabolism, you lose the anti-inflammatory effect.
Yeah, just on this point, I didn't get how this works. So the rewiring and the metabolic changes.
So to give you a short answer, it depends. So when you are thinking about host-microbe interaction, you have mechanism on the bacteria side and on the host side. So on the bacteria, we have ideas, but we are not yet sure about the molecule responsible for the effect. We have many, few hypotheses. We think, actually we are convinced that these molecules are present on the bacterial wall. So that I would say is the bacterial mechanism. On the host part, we think also we have identified some receptors. We are not yet sure about, not 100% sure, but we have some hypotheses. And what we see is that this activation actually push the energy metabolism toward oxidative phosphorylation instead of glycolysis. And this is particularly seen when you activate the cell. So if you stimulate macrophages with LPS, for example, they are going to glycolysis. But if in parallel you put this bacteria, this goes to oxidative phosphorylation, and then this is associated with all the anti-inflammatory phenotype.
So are there any attempts to sort of go out of this crazy difficult system and reconstruct it in a much simpler microbe?
So yes, of course, what we try is to identify the active component. If we find the active component, obviously we can use it directly. The more and more we work on these bacteria, the more and more we think there are multiple active components. So that's the first thing. The second thing is that, and actually here there is Jean-Marc Châtel in the room, who is working also on these bacteria since many years. We are not able to manipulate genetically these bacteria. And we are obviously not the only one to try in the world. So obviously, if we were able, to do transposon or this kind of type of experiment, it would accelerate a lot our discovery. But until now, it did not work.
I have a question that relates a bit to this, which is about can you comment on the genetic diversity of this bacterium? You say it could be up to 5%. I anticipate that there might be actually many different strains or subspecies, and do they all behave the same in terms of those effects?
Yeah. No, so there have been changes in taxonomy. Bacteriologists like to change the name and change the taxonomy regularly. So there have been, for some good reason, usually, it's not a criticism. And actually now there are even different species into the Faecalibacterium genera. They are all very close, but of course when you compare the genome, I'm not an expert in this aspect, but what I saw is that there is some common aspect. For example, they are producing butyrate very strongly. But what I can also tell you is when you test the immunomodulatory effect, they don't have all the same immunomodulatory effect. So there is some kind of heterogeneity. So if there is no more question, we are going to move to the second part of the short talk.